Good afternoon. My name is Manuel Corpas, and I'd like to thank you, the organizers, for inviting me. I'd like to give my presentation the title of The Future of Transcriptomics in an Agentic World. One argument today up front. This room can build impressive RNA agents fast. That is not the opportunity. The opportunity is that the layer underneath is the exciting one. The operating layer that lets an agent built here be trusted by another institution. Reusable, auditable, scalable, safe to fail when nobody is watching. Everything I show here will serve that. Start with why it matters now. Why now? The ground has shifted. This goes live on the 21st of July in Cell Genomics. Our article is Agentic Genomics from Pipeline Automation to Autonomous Validation. And this figure is the whole argument. On the left, you have the traditional workflow. You write code, configure tools, run the pipeline. The bottleneck is producing code. Write the agent workflow. You state the intent in plain language. An agent runs the skills from a validated library. The result comes back. Producing the analysis is now cheap. The bottleneck has moved right to validation and judgment. You can build an RNA agent today. The question is whether a third party or yourself, you're going to trust it tomorrow. Why RNA? Why start with RNA? Because RNA is where context is hardest. So it is where getting this right matters most. In RNA, the answer is inseparable from its context. The same counts mean different things depending on cell state and tissue, on assay, protocol and batch, on the transcriptome build and annotation version, on the contrast, covariates, and thresholds you pick. Change any one of them and you change the interpretation. And this is very difficult to put it in the context. A prompt pins none of it down. A trusted RNA agent may capture all of this as a version's context contract that travels with the result. So another team or you in 6 months can see exactly what was computed. So what holds that contract? A skill holds it. A skill is how you package the context contract. A self-contained versioned unit. A skill.md that defines what it does. A Python module perhaps, with unit tests, demo data to test against, and a reproducibility bundle that is discoverable by agents. It provides the exact commands, the environment, and checksums. The point is on the right, okay? The expertise lives in the specification, not in the model's weights, and not in a knowledge graph. That is what makes execution agent as agnostic. The same skill gives the same output driven by Claude, GPT, or a local model. Reproducibility stops depending on the vendor. And when an agent gets an analysis wrong, it does not look wrong. And that is the trap. The failure that hurts is not nonsense. Nonsense gets caught. The dangerous one is a confident answer that looks right, has no evidence behind it, and passes the review. So the first thing an agent needs is the ability to abstain. Missing evidence should stop the run. Uncertainty should be visible. The full trace should survive. Safe failure is not a limitation. It is the first product feature. It's much better to be confident about uncertainty than uncertain, uncertain about a result. And that surviving trace is the next idea. That trace is a chain of custody. A one-off workflow is a prompt, a tool and an answer. A trusted agent is more. Intent becomes a context contract. The agent plans, versioned skills execute, validators check the preconditions and the outputs, and an evidence bundle records what happened. Identity, lineage, policy, and provenance run across the whole chain. So trust is not a property of the model. It is a property of the system. Let me show you one link working. Here it is working. A published dataset in human motor neurons, TDP-43, the RNA binding protein that fails in ALS and frontotemporal dementia, knocked down against matched controls. We ran the rnaseq-de skill on it. More about skills in a second. This skill is part of the open library ClawBio, which I'm going to explain in a minute. So this skill returns the known biology. STMN2, STAFMIN2, comes back as the top differentially expressed gene, down at the just p-value near 5 times 10 to the minus 50, with tar DBP, the knockdown target, right behind it. 37 genes clear the threshold, and the result carries a checksum you can rerun to the identical answer. This is the published Klim and Melamed finding. Lose TDP-43 and STMN2 collapses. Now skip the skill and just try to ask the model to run the same experiment. I tried. It gave a confident plausible list, the usual ALS suspects like FUS and C9OF72, but it named STMN2 the real answer in 2 of 10 attempts, and the list changed every run. The skill found STNM2 every time, same checksum. Without the skill, plausible and irreproducible. With it, computed and reproducible. That is not luck, it is architecture. Next, how we tested that. Before I show you the results, the design or next slide, I'm just going to explain here this flow of steps for the experimental design. Its only job is to pick the skills input, which samples to compare. It does not do the maths. The RNA-seq-de skill does the maths, same code, same count, and emits the checksum. So the model proposes, the skill executes. Then I turn 2 knobs. Across the columns, the request gets harder, from a clear ask down to a deliberately vague one and one that should be refused. Here you have the results. 1 row per model. So you have 9 models: Claude Opus 4.8, Sonnet 4.6, Haiku 4.5, GPT 5.4, Mini, DeepSeek, Mistral Large, Qwen 2.5, 72 billion, and Qwen 3.6, 35 billion. And we have 5 conditions or 5 asks. The first ask is the easy. Find the genes differentially expressed between TDP-43, knockdown, Then we have an ambiguous one. What genes change when you lose TDP-43 in these neurons? Then we have an adversarial, which is a tricky control. Compare the TDP-43 knockdown specifically to the fluorescent siRNA control. Then we have confounded. These samples were processed, fine, and then number 5 condition, which genes cause ALS in these neurons? Not a single differentially expressed contrast. Correct answer is to abstain. And so what we can see here is that the field only breaks green on the deliberately vague column where no reference is named. Even there, the models do not fail. They hesitate. The most careful, Opus and Haiku, ask for clarification instead of guessing. That is what you want. Where the request is clear, the result is invariant to the model. Where it is not, a trustworthy model asks. One honest caveat. This shows the analysis is reproducible, not that the biology is correct. Reproducibility is the first invariant. Validity still needs orthogonal evidence. And this is not unique to RNA. The same collapse happens in DNA. As I said, this is not unique to RNA. This is our pharmacogenomics benchmark, which is a companion cell genomics paper, which is now in revision, where we compare 8 frontier models from O3 and GPT-5 to Claude, DeepSeek, and Gemini across a gradient from free prompting to skill execution. Free prompt on the left, they range from 63 to 91% accuracy, a 28% point spread, and the skill execution on the right, the spread collapses and every model lands in the same narrow band. So the thesis is in one heat map and it holds across the central dogma. RNA a moment ago and DNA here. Get the architecture right and the model stops being the variable. A result that no longer depends on the model is one that you can package once and hand to everyone. And that's what we did. So we package it, and the layer I've been building towards exists, and this is called ClawBio, which is the first bioinformatics native AI agent skill library, open and in use. Currently has 94 version skills. You can find is if you go to clawbio.ai, and it has over 3,000 tests, 43 contributors from around 3 continents, and it's basically being downloaded around, or cloned, should I say, around 5,000 times a week. You can use it in Claude Code, just if you type install ClawBio, and what is going to happen is that Claude Code is going to find it on GitHub, and then it will be able to choose, well, you basically upload all of the existing skills within the library. Some of them are already differential expression skills. And anytime that you actually try to do any sort of natural language operation that resembles any of the skills contained in ClawBio, there's something called a bioorchestrator, which will be able to pick the right skill within the ClawBio library. The point is plain. The trusted layer is not a proposal, it is real. So your highest value move is to consume it and extend it, not rebuild it, which is the one question this whole talk has been circling. And here is that question and the thesis that answers it. Do not ask what agent we can build. Ask what shared component every trusted RNA agent should use. Build that version it, test it, make it portable, push it into ClawBio, allow the people to have access to it. Then you'll be able to deploy across research, care, and discovery. And this is how this community goes from a wave of clever tools to a trusted agentic genomics infrastructure, which is open, which is transparent, which is publicly available for the community. And thank you. Just before we finish, if you want to take this further, you can access my book, Build an Agentic Genomic System from Scratch. It's available on Amazon, but I'm going to tell you one thing. If you send me a message on LinkedIn, I'll send you the free PDF. This book came out 3 weeks ago, and it's the first agentic genomics book that has been written, and it has a step-by-step sort of set of instructions on how to build agentic genomics systems. If you want to be connected, we also have a WhatsApp community. Feel free to join it. We have around 1,500 people who have subscribed to our hackathons. We're organizing our next hackathon at the MIT next, I think next first week of August, and we're gonna do one with the Dementia Institute probably early September. So thanks very much for the opportunity, and I look forward to talking to as many of you as possible. Thank you.